Hello and welcome to the In a Sense channel. Have you ever felt your brain working? Probably not. The closest may be the type of pain in tissues around your brain you felt during a headache. Well, today I'm joined by Dr. Rainer Gutiérrez, who studies the phenomena of appetite. One of the pioneering techniques he uses to do this is called optogenetics, and it involves stimulating brain cells with light. His lab has discovered a surprising side effect of optogenetics, which he proposes is a novel form of internal sensing, like interoception of the brain. His findings have some really intriguing implications for other researchers using optogenetic perturbations in their work, and after speaking to Dr. Gutiérrez, I was left wondering about how we normally learn to associate unusual patterns in our body with events outside of us. I won't reveal any more, but I hope you enjoy this fascinating interview with Dr. Rainer Gutiérrez, and please do subscribe to the In a Sense channel for more information about how we sense our inner world and how you can apply inner body awareness techniques to improve your overall well-being and performance. Thank you. Hello, Dr. Rainer Gutiérrez. Thank you so much for joining us here on the In a Sense channel. I'm really looking forward to covering this topic today.
Thank you, Ian, for the invitation. I'm really happy to be here.
So we've got a really fascinating new topic which hardly anyone will have heard about before. But before we move on to that, can you tell me a little bit about your background and how you ended up working at the Laboratory of Neurobiology of Appetite at the National Polytechnic Institute in New Mexico?
Oh, thank you, Ian. I study psychology in Mexico City at National University of Mexico, and then I did a PhD in biomedical science also at UNAM in Mexico City, and after that I did a postdoc at Duke University in the U.S., where I learned a lot of techniques for electrophysiological recordings in freely moving animals and taste coding. And later I decided to initiate my own lab, and then I returned to Mexico City, where I start my laboratory of neurobiology of appetite to understand the neural circuits that controls feeding behavior, and that's the main goal of my lab. But and basically that's it. I have been there for more than 14 years now from now.
So what sort of different research topics do your lab cover then?
Okay, so we study many topics, mainly the neuronal circuits of appetite is our main topic, but it comes to our attention one phenomenon that we observe with the mice when we are doing optogenetics, which is a technique that we can use to stimulate and modulate or even inhibit some cells, particular cell types in the brain, using genetic modifications and introduction of a protein called channelrhodopsin and light. So now we can make the cells sensitive to light and activate those cells with a millisecond precision, right? So that technique is optogenetics and was invented by Carl Diderot in 2005. It's a very terrific, a game-changer technology that has allowed us to modulate the brain in very precise ways. So it has been a game changer to interrogate the neural circuits in health, in disease and in health conditions. So we start observing a phenomenon where the animals, we were wondering how the animal experience these optogenetic stimulations, what they are feeling when we perturb their brains. And we found that the animals actually can sense these brain perturbations. And we call this phenomenon optoception, because we think is a phenomenon that arises from stimulating the brain itself from inside the brain, and similar to how it's occurred with interoception, right?
Fascinating. So, so yeah, optoception is what we're talking about today. And just to make sure I've understood that correctly, so this technique was developed to kind of study brain activity, and in the same way that other maybe brain stimulation techniques in the past have been used with electrodes or whatever. But in this case, you're introducing a protein to be expressed in specific areas, and this is a light sensitive protein. Is it? Is that what you said?
That's right. Particular cell types. Yes.
And so I guess you have to target the cell types in some way to express this protein first, and it requires there to be a way to target those proteins. I guess they have to have a unique quality in order to get the gene to express there.
That's right. It depends on the gene that the cell types express. We can use certain adenovirus to specifically infect the cells and express the opsin protein just in those cells. And once that the cells are expressing the opsins, the next step for optogenetics is to stimulate with a light. So we need to introduce a fiber optic into the brain of the mice to shine some light. And with that, blue light, for example, as the most used protein for is channel rhodopsin, and is sensitive to blue light, right? So when we introduce the blue light to the brain, these cells that contains channel rhodopsin now will start generating action potentials. Every time that we turn the light, the cell will be activated. Or we can also silence the cells, inhibit the cells with another opsin, with another kind of opsin, right? And a different types of light. So but it's a game changer technology to understand the brain.
Because it's so precise, is that why? Because it's
so precise. In comparison with the electrical stimulation, where you stimulate the brain, all the cells around and even axons are also activated when you introduce an electrode, right? With optogenetics, you have a higher precision to modulate and to understand the brain, to interrogate what function this cell type might have, right? So that's why it's important for neuroscience in general. And this is an old technique in 2005, was invented by Carl Teiser.
Okay, and so I'm going to want to dig in a little bit more. So the opsins there, those rhodopsin and the other opsin family of molecules, they're normally found, I guess, in the eyes. But I guess, am I right that you can get them in other parts of the body as well, so in skin and in blood vessels?
Let me tell you, these opsins comes from. We don't have these opsins on the brain normally. Channel rhodopsin is not present. There's only rhodopsin in the eyes, in the cones. But this is a different kind of opsin, right? So our brain basically is not sensitive to light at all. If you introduce a light inside the brain, you will observe just thermal effects, but no activation of the neurons. So this channel rhodopsin comes from an algae, and the gene of the algae you can introduce that gene into the cells of the human, not the human, sorry. This is just for animals.
The mice.
Not for mice, exactly, for mammal cells. And then the cells will start expressing that opsin. And now the cell will be sensitive to light, right? So that's optogenetics. And with that technique, the promise of that technique is that we can somehow get a label of different types of cells depending on what gene they express, right? The cells express, for example, D1 gene that express for a protein called dopamine receptor 1, or D2, or GABA, which is a inhibitory neurotransmitter, or glutamate, which is an excitatory neurotransmitter, okay? So we can label the cells depending on the genes that they express, and we can selectively activate different cell types using optogenetics. So this is a revolutionary technology to understand how the brain functions.
Okay, fantastic.
How the brain works.
Really good. That's helped clarify a few things for me. Great. So maybe in your paper then, so you've taken this photosensitive molecule that's normally in algae, it's not the same type as expressed in our eyes, and you introduce it in specific parts of the brain based on cells which express different genes, maybe neurotransmitters. And so then you can excite specific parts of the brain.
Or silence them.
Or silence. So it's a bit like
a switch. So it might be like a switch. It's kind of a switch. So you can turn on and off particular cell types in your brain, wherever the experimenter turn on the light or turn off the light. You see, it's a very powerful technique to understand what these cell types do, what are their necessity and sufficiency or their physiological function in the brain. For example, if we turn on some specific cells that are in the lateral hypothalamus that express GABA, so if we turn on those cells, the animal will start eating voraciously, right? Every time we turn on the light, the animal will start feeding.
So, okay, so it is literally like a switch, and whether it's on or off, and you can see what happens, and presumably you're trying to mimic things that happen normally in everyday life for that mouse. So why would it suddenly decide to eat or not?
That's correct. That's
okay.
Correct.
So, great. And in your paper, what did you just discover then? Because as I understand it, this optogenetics has been used before, but you were looking at it from a different perspective.
Correct.
And what were the surprising things that you discovered, or the unexpected things?
So I was wondering in the lab, with my colleagues, how do the animal experience this brain manipulation, these brain perturbations? So when we activate with optogenetics different cell types, and we see that the animal do something, sometimes we see that the animal do nothing, right? Sometimes there's some parts of the brain that you activate and nothing happens that you can visually see. But we wonder, do the animal feels something when we perturb the brain? Can they detect it and learn to use it? So we try to answer that question, developing a behavioral task that we call optogenetic cue alternation task, where basically it's a very simple task. Because the mice cannot speak to us, they cannot tell us if they are feeling something, right? But we can train the animal to report, making a movement or doing some behavior to report if they feel something. So the strategy that we used was to develop this behavioral task where there's two zippers, pretty much in the frontal wall of a box. And the animal has to lick twice in the left zipper and to obtain 2 drops of sucrose, right? And then if they keep licking here, nothing else happens. So they have to move to alternate to the other zipper to get 2 more drops of sucrose. So once an animal learns to alternate to lick between zippers, just in the middle of the way of where the animal is heading towards the other zipper, we perturb the brain of the animal. And that perturbation has now some meaning, because if the animal detected, the instruction is that the animal has to change the head direction and return to the previously rewarded zipper to obtain 2 more drops of sucrose, 2 more rewards, okay? If the animal don't pay attention to the cue and continue the alternation, they will be punished by 2 air drops or by 2 air puffs.
Okay.
Which mice don't really like it, right? So
sorry, just to interrupt. So you've trained them previously to switch every 2 drops, go left, right, and now you want them to change their behavior. So instead of going left, right, you want them
they have to change direction and to return to the previously rewarded zipper. So the only way that the animal can do this task is if they feel something with the optogenetic stimulation. So they can report to us that they are feeling something, and they return. To our surprise, every site that we stimulate and we play with different cell types, GABAergic, glutamatergic, dopaminergic cells in multiple brain sites. For all the places that we tested, in all cases, the animals learn to report and switch the direction, right? So basically, the animal is telling us that they are feeling something, and using that feeling to solve the task, to obtain rewards and avoid punishment. Okay.
So would they, just so I completely understand this, would they in their own time have eventually figured this out without any interruption in the middle? Would they have eventually learned the new pattern if they got the punishment by trial and error?
No, there is no way, because we did the same with control animals that don't express the opsin proteins, and we stimulate with light these brains that are not sensitive to light. Okay, so what happened with these mice is that even after 130 sessions, one session per day, they could not solve the task. Okay, so it's not merely using the light or even a thermal effect induced by the laser to solve the task. The only way that the animals solve it is by feeling the perturbation of their own brain, of that particular cell types, and then using that information to return and obtain more rewards and avoid the punishment. Okay.
So even after 130 attempts, they didn't learn it through just random
and every session contains more like 100 trials, pretty much. So we're talking about a lot of experience, and the control animals could not learn it. So for us, that indicates that the animal is consciously perceiving the perturbation of their own brain, and it doesn't matter where we are perturbing. That
sounds like the important thing, that it doesn't matter where, because you'd expect that some sort of activity in some areas might, that's how our brain normally works, by some kind of activity leading to a motor action, let's say, or a change in behavior. So it seems independent of the brain area. They just needed some kind of buzz.
At least for the regions that we stimulate cortex, prefrontal cortex, and nucleus accumbens, even the cerebellum. We stimulate the cerebellum, which the people thought is not involved in conscious perception, right? So even the cerebellum induces a sensation that the animal is experiencing and can use it to solve the task. And what is very interesting from our work is that we were really surprised that even not only the activation of the cells, but also the silencing of the cells can be felt by the mice, right? So that's a pretty novel discovery. In few words, the mice seems to sense their own brain perturbations. So it's not innocuous, right? Besides their physiological function, because if we stimulate one part of the brain involved on feeding, for example, the animal will go and eat. But besides that, also are feeling something, the animal, that can be used to solve the task.
So it's different. Is it different than, the Pavlovian conditioning, where you get the dog and you ring the bell?
Well, it's pretty similar in that sense. They actually, in this 1965, Doty make a wonderful manuscript, published a manuscript in monkeys, where they stimulate electrically different parts of the brain. I think he tested pretty much like 60 different regions in the brain of the monkeys. And they asked the monkeys to report if they felt something, right? If they fail to report the stimulation, they will receive a electric shock in the tail, right? A punishment. So surprisingly, every single site that Doty stimulated in the brain of these monkeys was perceived and report it and learn very well. The monkeys learn to report it. So this phenomenon is also induced with electricity, not only with optogenetics. See?
So these really tiny little perturbations in your case, so it's very small, tiny areas, and it's a little bit of burst of activity. And, most people, and me included, up until recently, would have thought that we can't really detect brain activity, really. It's kind of a silent computer. Right. But this is saying maybe that it is possible to sense. Obviously, if people have a headache or some kind of,
Right. So let me clarify this point. Effects in the 60s, in the 70s, he trained monkeys to voluntarily modulate their own brain activity. So how they do that? They put an oscilloscope where they were recording one of the brain cells, and the oscilloscope starts showing the action potential of that particular cell. So, and they train the monkey to increase the activity of that cell. Somehow the monkeys can do it too, right? So they start increasing the firing rate of that particular cell that the experiment is randomly recording. It's still a mystery how the monkeys do it, can do it, but they can solve it, right? That doesn't mean that we are sensing or action potential. We don't have that high definition resolution of interoception perception of our own brain, right? So we don't feel the action potential, but indirectly we can feel. Indirectly we can feel like a headache, as you said, or something artificial, because the brain is being activated externally, right? Not with the under physiological conditions. So probably what is happening is that with optogenetics, one of the main advantages of optogenetics is that you activate one particular cell type simultaneously in synchronicity. So when you shine the light, all the cells that have the opsins are firing an action potential at the same time. That is not normally seen unless you have epilepsy, right? Under epilepsy conditions, you will have this synchronicity that somehow it affects consciousness. It goes away with consciousness when the synchronicity is too high, right? But maybe this is what is happening when the optogenetics is making the cells firing at the same time. The brain might sense that the homeostasis is broken, that something is not good, and so they can detect it. The animal can feel that their brain is being perturbed indirectly, right? So we are not feeling the action potentials per se, but we can feel something depending on the brain site. And this is also very important for optoception, because we were wondering what is feeling now, what the animal is feeling. Is feeling a generic sensation or a unique sensation from every part of the brain that we are stimulating? To answer that question, what we did was to stimulate in two different brain sites in one single mice. Okay, so we stimulate, for example, the nucleus accumbens, a brain region involved in reward, and also stimulate the cerebellum. Okay, so the animals were trained first to detect the stimulation in the nucleus accumbens, and they learned to report it. They learned to detect it and report and solve the task with the stimulation of the nucleus accumbens. And then after they learn, we switch the stimulation to the lateral cerebellum for the first time in these animals. So the hypothesis here was, if the animals are feeling the same in the neck that in the lateral cerebellum, so the animal will continue performing the task without any problem. But to our surprise, when we switch to the lateral cerebellum, the performance dropped.
So it's not a generic sensation.
Feeling different sensation, a different qualia, right? Sensory qualia, right? It's experiencing something different in the cerebellum than they are feeling in the neck. But the animal can learn. If we continue stimulating the cerebellum, then the animal can learn that also to detect the cerebellum stimulation after 20 sessions. Okay.
So is it quicker the second time?
No, it's not quicker. There is no saving on the learning. We were expecting some kind of saving, but we observed no saving on the learning. But what is very surprising is that after they learn to detect both sides, we can, in one single session, interleave trials randomly, one from NAC, one from cerebellum. And surprisingly, the mice can now solve the task. It doesn't matter if it's from cerebellum or from NAC. So if they learn to detect these two sides, now they can use it.
You can use either.
They can use either, exactly. But this experiment is really important because it suggests that optoception is not a genetic feeling, a genetic interoceptive sensation, okay? Is unique. It depends on the brain site and the cell types that you are stimulating. In this sense, we also did another experiment. We asked the mice if they can distinguish different frequencies of the stimulation of the same brain region. Okay. So in one brain region, we trained the mice to tell us, are you sensing 10 hertz of stimulation of the lights versus 20 hertz of stimulation? If you feel 10 hertz, go to this port. And if you feel 20 hertz, go to the right port. And the animal can do the classification, right? So in one single brain region and one single cell type, the animal can distinguish a different sensation from 10 hertz versus 20 hertz, right? That means that I imagine this like when we stimulate different parts of the brain, is like playing a piano, right? With optogenetics, we can activate only the white keys or the black keys, okay, at the same times. But what happened with optogenetics? The noise that it produces with optogenetics is like tan, tan, tan. It's very raw, right? Because all the keys are being stimulated at the same time. It's not like a spatiotemporal activation that normally occurs during our brain to make music, right? Like a beautiful music. With optogenetics, you are stimulating just one similar cell type simultaneously. And that is the noise, something that is not physiological, that the brain itself is sensing that is not normal. It's artificial, right?
Yes. Although with the different frequencies you say, there is some sensitivity.
So what happened is that the animal can distinguish 10 hertz versus 20 hertz, and the sensation that it produces might be different, right? So the brain somehow is kind of like a piano, right? The music that it produces depends on the cell types, and that's why the optoception is not a generic sensation, a generic music. Is a unique sensation depending on how you stimulate the cells, right? And probably when you stimulate with electricity is different, the sensation that when you stimulate with optogenetics, right? Because with electricity, you activate the white and the black keys that are around the electrode, right? Surrounding the electrode. But with optogenetics, you only play the black or the white keys simultaneously.
So with those two different hertz, I would, from a simplistic point of view, I'd think if that region of the brain or those cluster of neurons is just like an on-off switch, then it shouldn't matter which hertz you do. So unless are you getting a stronger signal with one frequency than another?
Actually, that's correct. 20 hertz activates more cells than 10 hertz. Right? So it recruits more cells. Obviously, the feeling that produces might be different. And the animal is telling us that they feel different because they can distinguish.
It's not like they
versus 20 hertz.
It's not like they, with given the task, they for 10 hertz they go once to this, and 20 hertz they go, twice as often. They're actually going between two different choices.
Right. That's right.
Interesting. So what does this, I'm thinking about other researchers using optogenetics. I guess these findings are quite important. They'll now have to take into consideration, because they might be interpreting their results thinking there's something just, thinking it's only activity and that there's no other conscious awareness.
That's correct. So as I was telling you at the beginning, that optogenetic has been a game-changing technology, and it still is a very important technology that will help us to understand how the brain works in health and disease. Okay, but now we have to be more careful about our interpretation, especially when we study perception, right? Like, can we make a mice to experience, for example, let's say I'm an expert on taste, so I can speak about taste. This experiment hasn't been done, but how can we make the mice experience sweet when we stimulate the sweet taste receptor cell on our tongue? So if we activate with light somehow, or with optogenetics or any other technique that can somehow activate the cells that has the receptor for glucose or for sucrose, the animal will experience sweetness. Or if we activate the cells with the bitter receptors, the animal will experience bitterness. So that is a problem with optoception, because when we activate these cells, it might be that we are kind of recapitulating or mimicking the physiological function of the circuit, but it's not clear if the animal will experience sweetness or bitterness. You see, because it might be me feeling something very artificial, like very artificial that might look like sweetness, but not exactly sweetness, right? And that's what our optoception is telling us. Like the brain can sense that something non-physiological that's probably breaking the spiking hemostasis is being activating the brain, right? So the animal can learn from the stimulation, not only activating the physiological function, it's also feeling something. And probably that something is not natural. It might be artificial. Like, I don't know if you have tried it, but if you put a square battery, this is small batteries in your tongue, that you feel electricity, you pass electricity on your tongue. You don't experience sweetness, bitter, saltiness, and sourness. You experience something really weird, artificial, like it's called electrical taste.
Yes, like metal, but electric shock. Yeah.
Exactly. So maybe something like that we will feel when we perturb the brain with optogenetics, or even the sensor at the peripheries, right? Like the tongue. So that's why it's interesting, the optoception. Like it wasn't expected to see that every part of the brain that we play, that we stimulate, the animal can report that they feel something. I don't know exactly what they feel, because every part of the brain that we're stimulating produces a different sensation. Okay, so so that's the point.
Okay, great. And in our email communication before this interview, I mentioned the work of Mark Solms, and what when I thought about when I saw the abstract and looked at the overview of your paper, and I was thinking about, this is, if it is a new novel kind of sensation, a bit like the battery on the tongue you said, then it's adding an element of surprise to the mouse, and as you said, taking it out of homeostasis. And it got me thinking of the work of Mark Solms, which I've read recently about, and he asks some very interesting questions, such as why are we conscious of anything at all, and why isn't everything just relegated to automaticity? And then he introduces the idea of errors, prediction error, free energy minimization. And it got me sort of thinking about the parallels to what you're doing, really. You're sort of, this perturbation is maybe making the mice sort of hyperconscious, and
like
that's how
the noise of the piano, like boom.
Yes, yeah.
Like something is outside the hemostasis, right? like a not a normal music, like right?
Yeah.
And that's what we are doing with optogenetics. Actually, that's why it's called optogenetic perturbations, because we are not like now confident that we are reactivating the brain in all their subtle spatiotemporal activations. And well, regarding the Mark Solms idea, I will have to recognize that I haven't read this Mark Solms before you mention it, and I'm glad you point me towards the work of Mark Solms. I wish I knew before I wrote the paper, but it's absolutely consistent with the idea of Mark Solms. Like when something deviate from homeostasis, from the set point, from the homeostasis, you feel a negative feeling, right? And you are aware of that. You produce awareness, like a conscious awareness of anything that is deviating from the homeostasis, because you are telling to the system something is wrong. You have to do something to return to the set point. And when you do something to return to the set point, you will feel like a positive feeling, right? Mark Solms was very influential by Jack Pensep, which I was aware, like an affective neuroscientist that study negative and positive valence, right? And so I completely agree with that idea. Like probably this is what is happening with optogenetics or any kind of brain perturbation that we produce, that we probably will develop in the future. Every time that we try to perturb the brain, that will deviate the signal from the homeostasis, and we will feel probably something negative, that something is not correct, that is something not natural. So we have to do something to return it. Otherwise, like if you continue doing, for example, if you activate the glutamatergic cells in synchronicity for too long, you trigger epilepsy, right? So that's a common side effect of overactivating the brain constantly in a rhythmic way, right?
And does that happen in mice if you do with optogenetics,
yes.
if you carry on for too long?
It's a very common side effect of optogenetics, right? When you activate glutamatergic cells particularly in a rhythmic way, and if that rhythm is sustained for too long, the homeostasis cannot return to normal baseline firing rate, and then epilepsy occurs. Right.
Fascinating.
So yes, Mark Solms' idea of consciousness, which consciousness is defined like any experience, right?
Like any experience that we feel, that we're aware of, notice.
It's nothing mystical. It's just like any experience, like experience the sweetness of sugar. That is conscious. It's a conscious awareness, right?
Amazing. What I'm also wondering there was you were talking. I was thinking about. So I do manual therapy, hands-on therapy, and I swap with lots of therapists. And there's a technique called cranial sacral therapy. I don't know if you've heard of that, or if have it.
I'm not aware. But
so it's a very gentle technique where the patient is, or the client is laying on the couch, and the therapist sort of just supports the skull and gently, so it encourages the patient to relax, so all the muscles at the back of the neck let go, and then they support the weight of the skull, and it doesn't really matter what the cranial sacral therapist explanation of it, because lots of people disagree with it, but often the patients report very pleasurable experiences, like they're floating, or and for me it suggests a return to homeostasis. So people who've had lots of stress, kind of headaches or pains in the body. It's used for infants who have distressed infants after maybe a difficult birth. But I'm just thinking and I've received the treatment, and from an interoception point of view, you can almost feel like a softening within the skull. So I'm just wondering,
It's like if they were escaping from the grand oppressor, the gravity.
Yes.
Right.
Yeah. Yes. Yeah. So maybe it's reducing all these errors, reducing all the perturbations and actually allowing a return to homeostasis.
Yes. So I'm not familiar with the technique, but if it's similar to what the astronauts are experiencing when they finally release from gravity and they experience this sensation of floating around, with there is no need of a bed, right? So everything is just one is floating around. The people that affects the reference of what the laws of physics means, right? When you are under zero gravity conditions, your brain is like in a different state. It's something weird, right? This is not normal. So you will be aware of your entire body, right? You will have a different experience, like probably mystical somehow.
Yes, yeah, because it's so unusual. Yeah,
it's so unusual, right?
And similar. I'm just thinking similar to flotation tanks. I don't know if you've heard of, or the Dead Sea, floating in. Yeah, an unusual experience. And that can either be pleasant or unpleasant. Sometimes it's pleasant.
Yes.
Okay, so I'm aware of the time. So is there anything else from your paper you'd like to share, or where the direction of future direction?
That's a good question. Thank you for answering that. I think one potential application of optoception, once that we understand that this phenomenon exists, and it has been actually seen from many years ago, from 65 with Doty work, and 2008, Karel Svoboda also observed that perturbing the somatosensory cortex, the animal can report that they are being stimulated, right? So in 2008, there was a paper in somatosensory. They thought it's just because the animal is feeling like a whisker in the rat and the mice is moving, right? But or they extend those findings to all brain regions. And that also, at least the regions that we tested, probably there's some regions that will not be perceived at all, or if the intensity is lower, they will not be induce an optoception. That has to be determined in future research. But the main possible application of this knowledge of optoception is to use it as a sensory new channel to introduce information to the brain, like using, for example, in a brain-computer interface. So instead of thinking, I'm going to induce a sweet sensation, I'm inducing an artificial sensation, but that can be used to introduce information to the brain, and the brain can sense it and use it to control external devices, right? So this will open a possibility to use it in brain-machine interfaces. Has multiple channels. So even the frequency can be used as one channel. The activation or the silencing or different brain regions can have different meanings. So we can introduce more information to the brain in the future. I don't see. That's how I see like potential uses of this optoception, but also it opens the way to study interoceptive sensations induced by the brain itself, right? So when you activate one particular part of the brain, what the animal is experiencing, what is the feeling that they are experiencing, that also opens a new way of experiments. For example, can you induce fever? Recently, there was a paper activating with particular cells in the hypothalamus that triggers fever, of Catherine Dulac work. They induce fever, right? So sometimes there is also another paper suggesting that you can activate the immune system. I mean, the animal will not see and move, and you will not see any effect, but the immune system is being triggered by the optogenetic stimulation of that part of the brain.
I think I saw, was that the one where they irritate, they first found out which parts of the brain were activated with different irritants, and then took the irritants away and used the optogenetics, and it replicated the immune reaction in different parts.
Exactly.
So I guess, you could use that to switch on or switch off.
Now you can switch on off the interoceptive. And even more interested now, we can use also optogenetics, not just to perturb the brain, but also the periphery. For example, the viscera, right? So we can stimulate enteroendocrinal cells and triggers this subconscious feeling of the gut and the brain. And supposedly the gut and the brain are communicating. What kind of dialogues this gut and the brain has? So we can now study those in more details with optogenetics, right? So, and we know now that the brain is sensing perturbations not only from the periphery, but also from the brain itself. So that's our vision of what will happen with optoception.
Great. I like the first example you gave as well about the kind of maybe linking it to external devices, and the example you gave of the monkeys who learned with the oscilloscope, or they could influence this kind of biofeedback device. And
biofeedback. Right. The difference with EPFET's approach with volitional neuronal control of activity is that you have to provide an exteroceptive cue, a vision or a noise that tell the monkey that the cell is firing, right? Mm-hmm. With optoception, no need of exteroceptive cue. All is interoceptive. All comes from the brain.
Yeah. Wow. Fascinating. Great. So I could ask questions for several hours, but I'm aware we've only arranged to speak for an hour. And so where can people find out more about your work?
Basically, I have a ResearchGate. If you type my name, Rainer Gutiérrez, you will find all my research. And Google Scholar will also be useful. And I have a Twitter at Gutiérrez Rainer, like my last name and first name, where you can reach me.
Thank you very much.
Thank you, Ian. Thank you for this wonderful interview.
Yeah, I've thoroughly enjoyed it, and I'll add links to the Twitter, and maybe if I can get the ResearchGate link as well, so people can follow your work.
And they can actually download the manuscripts, most of the manuscripts, for free.
That's great. Thank you so much, Dr. Gutiérrez, and enjoy the rest of your day in Mexico City.
Thank you very much, and thank you to your audience.
Please remember to subscribe to the In a Sense channel for more information about the role of your bodily sensations and tips for using bodily awareness to improve your well-being.